All right. Well, welcome everyone to our weekly Friday seminar. And it's my real honor to be introducing Dr. Hans Paerl as our speaker today. Hans is the Kenan Distinguished Professor of Marine and Environmental Sciences at UNC Chapel Hill and the Marine Science Institute there. And he has been, as he described, for more than 40 years. He did his bachelor's, his master's, and his PhD all at UC California, Davis, and his PhD with Charles Goldman, who's one of the best known limnologists of the 20th century. Hans is really a remarkable scientist, and I'm going to wax poetic just for a moment. Indulge me on this, Hans. He has won some very major awards. He's a fellow of the American Geophysical Union, which is the highest distinction the AGU gives. He won the Odum Lifetime Achievement Award from the Coastal and Estuarine Research Federation. He won the Evelyn Hutchinson Award from the for excellence from the American Society of Limnology and Oceanography. He's published more than close to 400 papers since 1972. Those have been cited close to 60,000 times. 15 papers in Nature and Science, 10 papers with over 1,000 citations. He's going to focus today on cyanohas, but he's really done a tremendous amount of work in all sorts of coastal estuarine and freshwater systems on looking at nutrient cycles and their impacts. And he's really, for me, been a very inspirational scientist. I remember doing my PhD, 2 of his papers were some of the most influential for me, and that was his 1988 paper in limnology and oceanography on nuisance phytoplankton blooms, and his 1997 paper where he was talking about really the first wave of people thinking about atmospheric deposition and groundwater as new nutrient sources. And Hans has been, he's really been a pioneer on a lot of subjects. In fact, he's published papers where we're still trying to chase down the subject today. So, for example, a lot of scientists today are trying to understand interactions between bacteria and cyanobacterial blooms. And I note it was in 1976 that Hans first started publishing on that. He published a paper in 87 on something known as dilution bioassays that people still use today. My lab is still using. He's started looking at interaction between cyanobacterial blooms and zooplankton back in the 80s. And again, a lot of people still chasing that down. And in 1991, published one of the first papers on sequencing the NIF-H gene to understand nitrogen fixation. And, that's something that is really, people are still looking at today. So, really just a very, very impressive body of work, and a super nice guy. Who's withstood attacks from other scientists and been proven right, including remember Joanne Burkholder coming after Hans Paerl, and in the end, Hans being right, fish kills were not due to Pfiesteria, and Schindler coming after Hans Paerl saying nitrogen is not important, and Hans being right again. So I guess there's something to having a long career, Hans. You stick around long enough, and you'll end up being proven right. So anyway, without further ado, I give you Dr. Hans Paerl.
Well, thanks, Chris. And I just want to add, you mentioned the NIF-H sequencing stuff. That was really, getting some very innovative graduate students to start doing that kind of work, and also collaborating with John Zayer, who was one of my old buddies and one of Charles's former graduate students as well. The one thing I want to mention before we get going on the cyanos is that I did my PhD work on Lake Tahoe, one of the clearest lakes, well, one of the clearest large lakes around. And now I'm working on lakes that you can practically walk on. So I consider myself having gone the whole trophic route. That's probably my greatest accomplishment. Anyway, and you see some of the pictures of some of those lakes right there. So today, I'd like to talk to you about what do we know about what's driving these cyanohab blooms? and try to sort of integrate human impacts, climatic impacts, and then in the end, sort of, go over what can we do about this, what knobs can we tweak, and where do we stand in terms of sort of management options for dealing with these blooms. And you see all the different things there on that opening slide that I'll be talking about, sort of a combination of human impacts and climatic impacts. And the other thing I wanted to mention is that the cyanos, as you can see at the bottom right-hand picture, or the bottom middle picture, are, they've been around for over 3 billion years. So they've had a lot of time to evolve, come up with strategies to counter climatic changes and geochemical changes on Earth. And so it should not be surprising why they can take advantage of the current trends that we're seeing on the globe. This is just, part of their playbook. So, just kind of keep that in mind, and we'll maybe end with a discussion about that. So let's see, how do I go forward? Let me try this. Okay, so just a little bit of review that most of you know already. The blooms that we're seeing, these harmful cyanobacterial blooms, they're symptomatic of both human and climatic alteration of aquatic environments. And that, of course, is linked to human expansion, urban, agricultural, and industrial growth, which, of course, is generating more nutrients. And I want to stress both nitrogen and phosphorus. We'll get to that later. But also hydrologic modifications play important roles. Things like damming rivers and reservoirs and withdrawing water in certain places for agricultural use, for example. And then lastly, climate change. I guess no talk is complete these days without talking about climate change, but it's pretty relevant to the expansion that we're seeing with cyanobacterial blooms. And the one thing we know about cyanobacterial blooms is there's really great documentation that these blooms are intensifying and spreading. I know there's a lot of controversy about some of the marine blooms that are generated offshore, but one thing you can be sure of is that the documentation on increased cyanobacterial blooms goes back at least a century or even more. And we'll get to that later too. So why are we worried about them? Again, I think most of you know this, but they produce secondary metabolites that can be toxic to a wide variety of organisms, ranging from zooplankton to humans. Hypoxia is often linked to the blooms as they die and sink to the bottom and consume. There's a lot of oxygen consumption, leading to fish kills often, and also shellfish kills, which are really more relevant because they can't go anywhere. They're just sort of trapped in these anoxic environments. Odor and taste problems. That's really an issue with regard to human use of the waters that are impacted by these blooms. They produce a variety of odors that are noxious, and many of you have probably smelled these blooms. And even sometimes when cyanobacteria grow on the sides of buildings, after a good rainfall or something, you can smell some of the geosmin and other compounds that are produced. So that's an issue with drinking water, obviously, but also recreational fishing use and overall sustainability of these waters are impacted by the blooms. Just a very quick summary of who they are. I'm not a taxonomist, so I tend to group things in very broad groups. But the cyanobacteria really can be kind of divided morphologically into 3 groups. The coccoid guys that are ovoid to single cells, but they're often in colonies like microcystis, which you see up there in the upper left-hand corner. These don't fix nitrogen, and we'll get back to that nitrogen fixation issue a little later. So they require nitrogen, either externally loaded or regenerated nitrogen to grow on. And that's an important thing to keep in mind from a management perspective. The second group are the filamentous non-heterocystis guys. They are essentially stacks of undifferentiated cells that are filamentous. Sometimes they have sheaths around them, like Lyngbia, for example. Some of those can fix nitrogen, but they're mostly benthic, the ones that fix nitrogen. Planktonic ones are pretty much dependent again on combined nitrogen. And then you got the third group, which are the filamentous ones that have these cells called heterocysts, and they are shown here as fluorescent cells. They are cells that are differentiated biochemically. There's no photosynthesis going on in these cells, but they can localize nitrogen fixation. So it's thought that this group here, the filamentous heterocystous ones, are the most highly evolved because they evolved in an oxygen-rich environment, and to a large extent, the cyanobacteria themselves are responsible for oxygen evolution on Earth, at least in the early days. So, before there was any oxygen evolution, of course, they could fix nitrogen as much as they wanted because there was no oxygen inhibition of that enzyme system. So those are the three groups. They all produce these toxic secondary metabolites. We have very little idea of why these metabolites are produced. And again, they're called toxic, largely because they negatively impact us and our resources. But to the cyanobacteria, they're probably not toxic at all. And there's a whole slew of different hypotheses about why these compounds are produced and what role they play in cyanobacterial growth. And we won't go there, but that's for another day. Okay, the other thing to emphasize is that, if I had given this lecture 50 years ago, we'd largely be talking about lakes and freshwater systems. But cyanobacteria are not really limited to freshwater. They now expand into estuarine systems, as you can see in those three middle slides, and even coastal seas, as you can see from the pictures in the Baltic. So salinity is not an issue, at least in terms of their expansion. Now, there are some taxa that are sensitive to higher salinity, and this is work from Pia Moisander. Some of you might know her. She's at UMass, I think at Dartmouth. And Pia was a PhD student of mine, and she did a lot of work with salinity tolerance of the different bloom formers. And you can see that it varies with the different taxa, where some, like nodularia, for example, can grow all the way at full strength seawater. But others, like Cylindrospermopsis, for example, craps out at a pretty low salinity. And then a lot of them are sort of in between. So there's plenty of room for expansion of cyanobacteria into more saline waters. And that's really what we're seeing in many places around the globe. What controls them? And I'm going to talk to you a bit about nutrients, which is an obvious driver of cyanobacterial blooms, linked to human activities mostly, excess nutrient loading, which of course provides the nutrient inoculum for them to grow in receiving water bodies. But they also are very sensitive to physical factors in the water body, including salinity, either temperature or salinity stratification. Residence time is really important. That is the amount of time that it takes for a drop of water entering a system to exit. Because cyanobacteria, despite the fact that they form these spectacular blooms, their growth rates are often much lower than other phytoplankton. So they just need longer residence time to be able to compete with other phytoplankton taxa in places where they are impacted by excess nutrient loading. And of course, once they're there, they can essentially move up and down the water column by altering their buoyancy. So during the day, often you'll see them bloom on the surface. And then at night, they can go down and get nutrients from the hypolimnion or even sediments. So they're very well adapted to taking advantage of both photosynthesis and nutrient retrieval. The other thing about blooms is that when they bloom at the surface, they shade underlying phytoplankton and even attached macrophytes. So, they're doing a pretty good job of outcompeting organisms that might have a faster growth rate, but if they don't have enough light to grow on, the cyanobacteria do just fine. And then lastly, they are, many of them are not grazed, or not grazed effectively because they grow in big colonies and filaments that are not easily grazed by zooplankton. And so, you know, the food web is altered very often by them in terms of taking, for example, rotifers that can take advantage of a bloom, but not a copepod or a cladostrum. So there are food web impacts there as well, beyond just the toxicity issue. Okay, I'm going to start with my favorite lake in China that I've been working on for quite a long time, Lake Tahoe. And Tahoe actually means large water in Mandarin. This is the third largest freshwater lake in China. It's about 2,300 square kilometers. It is a lake that changed from a oligo to mesotrophic lake back in the 60s, 70s, and even 80s. And then with the growth of major urban areas around the lake, this is Wuxi and Suzhou, each one of these cities now has about 8 million people in it. And even 10 years ago, they had like 2 million people. So the growth has just been phenomenal in terms of wastewater that's been generated by these cities. And then also agricultural activities have increased a lot in the watershed. And the Chinese, just like us, have learned all these bad things, like using chemical fertilizers instead of recycling organics, which they were doing 100 years ago, which was environmentally much better than what they're doing now. So with all that nutrient loading, the lake has really gone through a state change. It changed into essentially a cyanobacteria-dominated lake about maybe 15 years ago. And that happened really fast. The lake is dominated by microcystis, which is a non-nitrogen fixing surface bloom former. And here you can see me driving our vessel through there, sort of like driving through green whipped cream. The boat actually slows down when you're going through one of these blooms. And again, microcystis is a non-end fixer, so that's telling us something about nitrogen. We'll get back to that. These blooms last about 9 months out of the year now. We've done a lot of work with bioassays, which is something I learned from Charles a long time ago, except Charles was incubating them in a refrigerator. And, you know, all the stuff we do now is in situ, using natural light and temperature conditions and incubating outside. And we like to use these cubitainers, and they range from 1 liter to 20 liters. They're great to work with. They are PI proof, you know, you can't break them. You can even play a game of volleyball with them. And so we've used this in Tahoe to try to get an idea of what the nutrient limitation story looks like in Tahoe. And what you can see is that in the spring, so let's talk about the treatments first. This is initial lake water. So that's the water coming pumped out of the lake. The control with no nutrient additions, nitrogen added as nitrate, phosphorus added as phosphate, and then the combined additions of N and P. And what you can see is that there seems to be a sort of bimodal nutrient limitation phenomena going on in Tahoe, where phosphorus stimulates in the spring, but nitrogen assumes a more important role as a limiting factor in the summer. And here you can see the second year, again, P stimulation in the spring, N stimulation in the summer. That P stimulation is largely due to the fact that there's a tremendous amount of nitrogen coming from spring runoff into the lake. So the N to P ratios are astronomical, leading to P limitation. But that quickly goes away and things reverse to a more nitrogen limited situation as the bloom, of course, is sucking up all the nutrients out of the lake and nitrogen becomes limiting. And we'll get back to this in a second. But if you add N and P together, you get more than N or P alone, which is not uncommon these days. And I'll show you some other examples of that in lakes and estuarine and even some coastal systems. So both nutrients are important. They're important at different times of the year as limiting factors. That's the main thing I wanted you to get from that. This is my politician slide for politicians that don't like to look at data. And what you're looking at here is 100 mils of Tahoe water filtered from these bioassays, and looking at, you know, the color on the filters, the control, no nutrients, Nitrate. So this is a summer bioassay, by the way. Nitrate stimulates. Phosphate to some extent, but not as much as nitrogen. And N and P together give you more than N alone. So, it fits the actual data quite nicely. And for all practical purposes, this is what they need to be convinced anyway, that they need to do something about both nutrients. So the next question usually that comes up is, okay, you've convinced us that both N and P are important. How much do we need to reduce the inputs of these nutrients in order to get to a true sort of nutrient limited condition that hopefully will help mitigate the blooms? And so to address that, Chris already mentioned dilution bioassays. We do them in Tahoe. So instead of adding nutrients right away, we dilute the lake water with a dilution solution that has just the major ions in it, but no nitrogen or phosphorus. And so, you dilute serially here, and then you add each nutrient back individually to see which nutrient is limiting, okay, and under different dilution conditions. And when you do that in Tahoe, you get something like this. So P, for example, in order to really get P-limited growth in the spring, you need to reduce P by about 50% or so, 30 to 50% or so. For N, it ends up being even more, 50 to 70% for nitrogen. But the interesting thing, these numbers are big, obviously, but the interesting thing is if you look at the historic increases in loading of those two nutrients in the lake, it's pretty much paralleled what we're seeing in the bioassays. So, the nutrients have increased by at least those amounts in terms of loading. So the bioassays, I think, are pretty useful in terms of at least initially targeting what the reduction should be in order to really get nutrient limited conditions at different times of the year. Let's go to Lake Erie. And interestingly enough, Lake Erie is plagued with the same organism that dominates in Tahoe, Microcystis, several species, non-nitrogen fixer. Lake Erie in the 60s already started to develop cyanohab problems. And it turned out that, in the 60s, if you open up a limnology textbook, phosphorus was really the key nutrient that everyone was worried about in terms of nutrient limitation. And sure enough, phosphorus was reduced from these major wastewater treatment plants of cities around Erie, and they were able to control the bloom back in the 60s by only reducing phosphorus. Interesting story, because now the blooms have returned, or they've been really coming back over the last 10 years or so. And they're dominated by, instead of a nitrogen fixer, they're dominated by Microcystis, which is a nitrogen requirer. So this is already hinting that in addition to phosphorus, nitrogen is probably playing a really important role in terms of controlling these blooms. And if we do a bioassay, this is from 99, Sandusky Bay, which is an arm of Lake Erie, you can see that, and this was a summer bioassay, by the way, nitrate stimulates, ammonium stimulates, urea stimulates, phosphorus by itself, not really significant stimulation, but if you add N and P together, you get more than N alone or P alone. And here's the dilution results, okay, if we dilute by 40%. So again, nitrogen does play a really important role in these blooms in addition to phosphorus, but co-limitation is probably the key here, just like it is in Tahoe. The bioassay work that we and others have done has been criticized because it's in containers and doesn't really include sediment water column interactions, for example, to look at nutrient cycling. But really, they're meant, the bioassays are really meant to take kind of a snapshot of what's limiting at a certain time, and you can do them seasonally. But again, if you want to really get at the whole lake situation, you got to fertilize the whole lake. And, some folks have the luxury of being able to do that. And what you're looking at here is results from different experimental lake areas. They're mostly in the Northern Hemisphere, ELA in Canada, as well as in Quebec, Northwest Territories, and Sweden. The bottom line is if you fertilize with nitrogen, you get pretty good stimulation. With P, not so much, but there are some situations where you get stimulation. But if you add N and P together, you get more than N alone or P alone. So even the whole lake addition experiments are telling us very similar story to the smaller short-term bioassays that we've been running. And there seems to be a consistent story there then in terms of nutrient limitation. This is from a paper we published in ES&T with Wayne Wurtzbaugh and others that basically summarizes the results. Yes, P is important, N is important, but it takes two to tango essentially. N and P give you more than those nutrients alone. And I went ahead and summarized data from lakes around the world and also some estuarine places where these types of bioassays have been run. And overall, the results seem to mirror what I've shown you, that in most cases, the stimulation is greatest when you have N and P together. And this has really been summarized in lots of other good papers, Jim Elser, et cetera, Bill Lewis and Wayne Wurtzbaugh and Dan Conley, et cetera. So the story seems to be consistent. Both N and P are important. So, you know, from a management perspective, there are very few places now, in terms of lakes at least, and even some estuarine situations, where controlling only one nutrient is going to solve the problem. I think that's really the bottom line. Okay, this question is a good question. You know, if we start reducing nitrogen in these situations, like Erie and Tahoe, where microcystis and non-nitrogen fixers dominant, are we just going to convert, you know, the bloom from a non-nitrogen fixing harmful bloom to a nitrogen fixing bloom? And this has been, you know, this has been raised, this question. It's a good question. And we tested it in Tahoe by doing these larger mesocosm bioassays, where we added N, P in excess without adding N and P together, and then here's the lake water, blank lake water. And if you look at the nitrogen status of the water after making these additions, clearly it's going to increase with nitrate and N and P together. But interestingly enough, and I think you can see this, yeah. If you add P, you don't get any stimulation during this period when nitrogen's limiting. And, you know, if nitrogen fixation were really important in terms of catching up stoichiometrically to the phosphorus additions in this lake, you would see total N increase as well. And you don't see it, so it looks like nitrogen fixation, while it is can be an important process from an ecosystem perspective, it doesn't seem to really get over the problem of nitrogen limitation. So I think from this perspective, I think we probably can be fairly safe about, you know, reducing N without having to worry too much about the system getting taken over by N fixers. Maybe at a later stage, but certainly not early on tackling eutrophication. So why is this? Why don't we see enough nitrogen fixation to compensate for nitrogen limitation in these systems? And this really goes across to the marine situation as well. So here's the nitrogen fixing enzyme, the dinitrogenase reductase. Here's the phosphorus link. You got to have phosphate to produce ATP to power the process. So here's the phosphorus limitation issue with nitrogen fixation. But what I want to point out is that there are other factors that can limit this process, including the actual energy requirements. And in cyanobacteria, that would be through photosynthesis, but in bacteria through heterotrophy. Iron can play a potential role because iron is part of the enzyme system. And then lastly, don't forget about oxygen. Turns out this enzyme is very strongly inhibited by molecular oxygen. So all three of these then, plus phosphorus, are probably interactively controlling N2 fixation. And if you're really dealing with a turbid system like Tahoe or Erie, light limitation might actually play a pretty important role in terms of controlling the ability to fix nitrogen at a highly efficient rate to make up for the ecosystem demands. And getting back to oxygen, we did some interesting stuff in Tahoe with this. The oxygen problem. During blooms, you get O2 supersaturated conditions, and they could be potentially inhibitory to N fixation. And what this shows is that even in heterocystis cyanobacteria that have these special cells to protect the organism from oxygen, those are not perfect. Apparently there's inward diffusion of oxygen to inhibit the nitrogen fixing process. And this is really early work that was done by Peter Fay way back when, showing that with Anabina, if you increase the oxygen, you get to, here's the optimum N fixation. This is acetylene reduction, by the way, which is analog for nitrogen fixation. You see there's an optimum there about 0.2 atmospheres, but if you get into oxygen supersaturation, you actually start inhibiting the process of N fixation. So this is kind of an interesting paradox. You get these blooms that require nitrogen, and the nitrogen fixers are there, but they're also producing a ton of oxygen in these surface blooms. So in a way, they're kind of inhibiting their own ability to get around nitrogen fixation. And this is a very interesting evolutionary question, and also an important ecological question. And that may be one reason why we do get this chronic N limitation. If you look at budgets of N input by N fixation and the main loss mechanism of nitrogen by denitrification, these are all eutrophic lakes that have major cyanobacterial blooms on them. You see that the net flux is outward. So that means that denitrification is exceeding N fixation. You're losing more nitrogen from these systems than you can gain by N fixation. So nitrogen ends up being limiting, even with N fixation going on in these systems. And conceptually, this is how it works, at least in my mind, that we get the nutrients coming in. Black is nitrogen, orange is phosphorus. They support enhanced production, eutrophication, and ultimately blooms. The blooms then provide the carbon to power various processes, and one of those is denitrification, okay? Whereas nitrogen fixation is mainly controlled in the bloom itself. And so what you can see is that as you increase the trophic state, you're enhancing the ability to denitrify at the same time, and N fixation is not really impacted the same way. So as a result, and of course phosphorus, once it gets in, is not leaving very quickly because there are no gaseous forms for it to escape. So it's merely cycling around. There's some loss advectively, but it's pretty small compared to the inputs, whereas N is being vented by denitrification. So as a result, these systems are always nitrogen hungry. As they get more eutrophic, they're more nitrogen hungry. And what we're seeing now in the literature is that more eutrophic systems are more nitrogen limited. It's kind of a hard concept to grasp, but turns out that, if we pollute systems enough, they become more nitrogen limited over time. And that, of course, begs the subject of reducing nitrogen to control primary production. Okay, the second part of my talk deals really with the climate change issue. And what I'm going to talk about are really the main issues that we're concerned with in climate change. One of them is warming, obviously, and you can see the North Atlantic anomaly here over the last 100 years. And also more extreme rainfall and drought events. This is the tropical cyclone record. This is not something we like to see in Beaufort because we're right in the middle of all this stuff. But that's all increasing. So we're getting more increased major storm events, but also major droughts. And what I want to point out is that, the storms, of course, deliver the nutrients, but the droughts increase the residence time in the system after the nutrients have been delivered. So that's kind of the perfect storm scenario. You want to get your nutrients in there and then increase the residence time to optimize the cyanobacterial growth. Temperature is a key driver of lots of things, including growth rates in the phytoplankton. And what you can see here on the right is that if you plot temperature versus maximum growth rates, here are diatoms, here are dinos, chlorophytes. These are from cultures, by the way, that we've gotten out of the literature. And cyanobacteria, which opt out or maximize out at pretty high temperatures. So they like it hot, so to speak. The warmer it gets, the better they can compete growth rate-wise. So that's one plus. And we know that a lot of systems are increasing in terms of temperature in the epilimnia. This is data from the Netherlands from a wide variety of lakes, showing you the increased epilimnetic temperatures that we're seeing over the last 100 years. And even Lake Erie, even though the data are very scattered because you get, warm versus cooler spring, summertime issues and things like that, high variability, the net effect is that it does look like even Lake Erie is warming up in the epilimnion. So these trends then that we see definitely would benefit cyanobacteria just based on growth rates. And if you look at some field data, this is data from Sar and Kosten et al. From the Netherlands, where they took 143 lakes, monitored them all the way from Tierra del Fuego up to the Arctic Circle. And what they showed, very interestingly, is that as you encounter increased temperatures and increased TN, that's the combination that leads to the highest dominance of cyanobacteria in the biovolume of the phytoplankton. You can use TP as well, although it turns out the relationship is not as good as with TN, maybe for the reasons I've explained to you earlier. But this shows, in fact, that cyanos do like it hot. Now, not all these cyanos are cyanohabs. They're just the percentage of cyanobacteria in the total phytoplankton counts. But I think it's pretty compelling evidence that temperature and temperature increases are playing an important role, and that nitrogen is also playing an important role. So going back to Erie, interestingly enough, Erie is, of course, the warmest of the Great Lakes because it's the shallowest and smallest volume-wise. So it may be that what we're seeing in Erie is a combination of increased nutrient loading, but also increased temperature that might be favoring the combined, or the combined effects might be favoring more enhanced cyanobacterial bloom potential, particularly in the western part of the lake, which is very shallow and warmest. If we stay with Erie, you can see that the cyanohab problem is increasing in severity over time. Not too surprising. I think many of you have seen these plots already, these trendy plots. And from the report that this comes from, the blooms appear to be starting earlier and ending later, and that can be shown pretty well with first day of bloom. If we use 17 degrees as kind of a threshold level for blooms to form, the first day of blooms versus the last day of blooms, those are increasing. The window is increasing between the first day and the last day. And that's leading to more annual growth days above 17 degrees centigrade. And you can see that here in the plot that the growth dates, the window of opportunity is definitely increasing in Erie. So there are trends there that can be teased out to show that. So for Erie, the changes in water temperature patterns are leading to a longer bloom window. They start earlier and last longer. Increased growth potential could lead to blooms intensifying earlier, and work by, among others, Chris and his crew shown that the toxicity potential is quite high in the spring because there's a lot of nitrogen sitting around to help synthesize those toxins. So the toxicity may be increasing as well, simply because the window is overlapping with when there's a lot of nitrogen in the water. And so this leads to an increased risk for water quality managers and recreational uses of those waters. And temperature, while it is important, it's not the only factor driving this increase in blooms. And that gets me to the last part of the talk, which is hydrology. Things are getting more extreme in terms of rainfall, but also droughts. And that's certainly impacting many systems that we're concerned with globally. In Erie, of course, the watershed is getting more extreme rainfall events, which means, of course, more nutrient inputs to support the blooms. We also have a similar problem in North Carolina. This is some data, or just some visual data from the brackish Albemarle Sound, which is the northernmost part of the Albemarle-Pamlico Sound system, the second largest estuarine complex in the US. Albemarle Sound is brackish because it's cut off from the ocean by the Outer Banks, and the water flows into Pamlico Sound to the south. And what we're seeing with more storm events and episodic events is that these blooms are moving beyond the rivers on into into Albemarle Sound itself. So this increased sort of episodicity of rainfall events is obviously leading to more nutrient inputs, but also freshening up the system and moving things into water bodies where the blooms haven't been reported thus far. North Carolina is not the only place where I can show you examples of this. If we go to the northern Gulf of Mexico, the receiving waters for the Mississippi and Atchafalaya rivers, and this is work by Sybil Bargu, by the way, and her colleagues at LSU. They have shown that floodwater-driven blooms are going further and further into the coastal waters because the waters are freshening, but they're also getting more nutrients. And as a result, you're seeing that the composition is changing, the more cyanobacteria, and of course the toxicity associated with those blooms is increasing as well. So, you know, the blooms are going well beyond their typical freshwater habitat and moving into places where they have not been reported up until recently. If we go to Florida and Lake Okeechobee, which has been a cyanohab reactor for a long time, Okeechobee is actually not a lake. It's a reservoir that's managed by the Army Corps of Engineers, and it has lots of levees around it and stuff. And of course, with the more extreme events that are happening also in Florida, they have to relieve the water out of Okeechobee or they'll break their levees. And so what's happening there is that the bloom is actually being transported down the Caloosahatchee to the Indian River Lagoon area, but also to the Gulf of Mexico. And as a result, you're getting transport essentially of what used to only be a freshwater problem into estuaries. And some of those blooms can survive for some appreciable amount of time. This is, you know, multi-million-dollar houses now with blooms in front of them. And if the blooms die, of course, the toxin will be released into the water. So it ain't over when it's over in terms of water quality issues with these blooms. Blooms don't necessarily go away when we have big storm events, or at least they only go away for a short period of time. This is data from the New River Estuary in North Carolina. It's just, it's about halfway between where we are up in Morehead City and Wilmington down here. This is a small river that is bounded by a bar-built barrier island out here. So the residence time is pretty long, of several months or so in the New River Estuary. And we've deployed vertical profilers there to look at temperature, salinity, dissolved oxygen, chlorophyll, and turbidity. And what I'm going to show you is we had the good fortune of having a tropical storm impact the area that didn't wash out our vertical profiler. In fact, the vertical profiler did just fine during the storm. And you can see here's the wind field of the storm and the mixing that occurred. By the way, this estuary is very shallow, only 3 meters, 3 to 4 meters. So temperature is uniform, salinity is uniform, DO, chlorophyll, of course, is spread throughout the water column. And here's turbidity. And this turbidity is really sediment resuspension that's going on in the system during a storm event. And what I want you to focus on is the chlorophyll. This is the bloom that was starting to develop. And then the storm came along, mixed everything up. And look at what happened after the storm. The bloom came back at a greater magnitude than it happened before the storm event. And what we think is going on here is that the storm resuspended sediments with nutrients in the water column. And of course, also the runoff associated with the storm would add nutrients. So cyanos like storms, as long as they're in long residence time systems where they can grow back and take advantage of them, okay? And if we go back to Tahoe, I've been there enough times to actually see lots of typhoons and what happens after them. Each arrow is a typhoon that occurred, actually they occur very much. You can set the calendar by them. They usually occur in mid-August there if they're going to occur. So here's 3 typhoons that occurred. This is MODIS chlorophyll A data in the lake. And you can see after each typhoon, the bloom actually came back to a greater extent than it was before the storm event. And we think it's a very similar phenomena that sediment resuspension and also runoff is supporting the continued bloom activity. And, you know, August is sort of midsummer in China. There's still plenty of warm, sunny days left for the bloom to reestablish itself after the storm occurs in Tahoe. In fact, the blooms don't go away until usually November or December. So same kind of phenomena. Here's a typhoon-free year. So you can see that it's probably more chronically nutrient limited. But here the nutrients are made available through the storm itself. And so the challenge in Tahoe is, you know, we're getting increased temperature, particularly at nighttime. The wind speed has actually decreased in the Tahoe Basin. This is long-term data from WUSHI showing the average wind speed has decreased. So that has increased the potential for vertical stratification. And of course, blooms like stratified column conditions. Rainfall has increased. So we're getting more nutrients coming in from rainfall. And here you can see the relationship between wind speed and chlorophyll in the lake itself. So we don't only have the challenge of trying to figure out how much nutrients need to be reduced, but how much more they might need to be reduced when we have climatic conditions that are setting the bar lower for the bloom to form. So this is, you know, kind of a dual challenge. It's, you know, we can tell you what the nutrient reductions need to be with a bioassay, but that might change over time as these climate change conditions increase the potential for blooms to form. So just summing up, what do we got to do about all this? Well, it looks like in many cases we got to deal with both N and P. I don't think it's feasible anymore in many places to only reduce one nutrient to control these blooms. You've got situations where, you know, P loading has been going on for 50 years or longer with a huge legacy of phosphorus in the lakes, which seems to be linked to nitrogen limitation being more common. So got to reduce both to really have some controlling effect. Those thresholds are probably system specific, but in many cases where we've done bioassays, it looks like we need to deal with at least a 30% reduction for both nutrients. That should be the target, at least anyway. Salinity is not necessarily a barrier to the expansion. And, you know, we could be, maybe not in my lifetime, but beyond, we could be seeing these blooms appearing in coastal waters if they're nutrient enriched enough. We're seeing some glimpse of that already Of course, the Baltic is a really good example, but there are other situations where we're seeing that now. Northern Gulf of Mexico, Florida Bay, other places. We may need to reduce N&P inputs more in a warmer, stormier world because blooms like it hot, and these episodic events seem to the blooms if you get a flood situation followed by a drought. And then lastly, the nutrient input restrictions really need to be year round. I've been asked this question a lot in China, well, okay, can we just reduce P in the spring and N in the summer? That's not going to work because many of these systems have long residence time and a long memory and a long legacy of these nutrient loading. So, I think we're in for restrictions that are on a year-round basis. And lastly, the window of opportunity is increasing, particularly in high latitude areas where earlier ice off occurs and later ice on. So, really the window in terms of optimal light conditions and nutrient inputs is increasing. And so I think I'll just leave it there and see if there are any questions. Chris, I don't know if I used up all my time, but
No, we have plenty of time for questions. So, and we've got
as long as you guys can tolerate me.
And well, I know some students are going to meet. I have set up a student meeting for after this, but for right now, attendees can ask questions. I see Gordon Taylor has his hand up. So Gordon, go ahead.
Okay. Hey, Hans,
Gordon.
thanks a lot. How you doing, buddy?
Good. Good.
So you brought up a point about hydraulic residence being so important for the cyanobacterial blooms, largely because they grow more slowly than microalgae. My question is, why is that? Why are they slower than microalgae?
Well, they're bacteria, and I think I think it's related to optimal temperatures. I think like bacteria, their temperature optimum is usually in the high 20s or maybe even low 30s. So, if you have a spring bloom kind of situation, those diatoms that you got in the background of your nice picture there are gonna, they're gonna do much better than cyanobacteria because their temperature optimum is, is there. And so, in order for them to compete, they need longer residence time because they got to have that time to how they do their other mischief, like shading underlying algal populations, for example.
Okay. Yeah.
Maybe having some impact with the secondary metabolites they produce. There are many strategies we haven't even uncovered, I think, that are implicated in that.
Well, I know from the literature that like Q10s for most photosynthetic organisms are on the order of 2.0, and heterotrophic bacteria at least are more like 3. So they are way more temperature sensitive. I didn't think that applied to cyanos as though.
Well, they are bacteria. They're prokaryotes.
Oh, I know. Yeah.
Yeah. So they haven't.
But photosynthetic, I thought it. Yeah.
Yeah, they haven't outgrown that capability of being a prokaryote. But just wait and see.
Okay, I'll give them time. Thanks.
It's sort of like the eggplant. That ate Chicago. Anyway.
Thanks, Hans.
Right.
So I think that's their, I think that's their thing. I think that if you look at phytoplankton successional data in lakes and some estuaries, the cyanobacteria don't really come up. They don't really. They're not really an appreciable segment of the biomass until you get above about 17 or 18 degrees.
Yeah.
And that includes the picoplankton too, by the way. The picocyanos,
Right.
They fall into that same kind of temperature, that linkage to temperature.
Cool. Thanks.
Okay, other questions?
Cody Garrison is.
Cody, go ahead.
Hello.
Hi, Cody.
Hey, Hans. Great talk. I was just curious what you might think about the residence times and how they might change in the future, specifically in that North Carolina system with rising sea level and more extreme storm events and everything, all the different effects. Do you expect that residence time to decrease or not be as long in the coming years and decades?
Yeah, no, that's a good question with regard to sea level rise. I really don't know. I think, the Albemarle Sound has been fresh for a long time. It's interesting because those the barrier islands are so thin, and you would think that they would get overrun historically by hurricanes that would form new inlets and things like that.
Right.
It has happened in Pamlico Sound, and Pamlico Sound is definitely more saline. It's about half seawater salinity, well, 2/3 seawater salinity. So it could be that, a major storm will come along that will make the Albemarle Sound more salty and also reduce the residence time. We just haven't gotten there yet, I think. Hopefully it won't occur in my lifetime because I think we'll be underwater in Beaufort. But,
right.
Yeah, the Gulf of Mexico situation with Sybil's work is interesting because there you have almost an infinite residence time, once you get into the northern Gulf of Mexico. Well, it's just going to be a question of salinity and nutrients in terms of how long these blooms can be there and last. So we are seeing much longer residence time in lakes out west for sure. I've been doing some advisory work in Utah Lake, for example, which has really huge problems with these blooms. And of course, the extended droughts there are increasing the residence time as well. And that's part of why they're having such big problems, even with nutrient reductions.
Great, thank you.
Okay. Thank you, Cody. Let's go to Bob Aller.
Hey, Bob.
Hey, Hans.
How you
Very
doing?
stimulating presentation. Thank you. And you obviously have too much time to do research. So, yeah.
Well, that's what, yeah, I've been hiding out at Moorhead for this long, but, we got merged with 2 departments on campus, so now they want me to teach 3 courses a year.
There you go. That'll do it. That's the end of you.
Well, I think that'll be the message to my retirement early.
Good. Okay. My question concerns the interaction between these organisms and nitrifiers. Because you were talking about denitrification as an important process in these systems, and of course you need nitrate to carry out dinitrogen production by whatever pathway. And so I'm interested in how the HABs affect the nitrifiers.
Yeah. Well, the nitrification is a relatively short, slow process, right? Compared to the other steps in the nitrogen cycle. And it could be that is the limiting factor in terms of how much nitrate you can produce by regeneration in these systems. But the problem is that we're getting so much nitrate loading from external sources in these systems that nitrification may not be as critical as it would be, say, in a more oligotrophic system, or one where you would have to depend on regeneration. And we're getting a lot of groundwater input that's pretty high nitrate too. So whereas nitrification could be a choke point, for denitrification, that's for sure. But we're seeing so many systems where there's plenty of nitrate available that kind of overrides it.
Okay. Can I ask one more question before we pass, I guess, to Malcolm, which is,
Sure.
Please, Bob.
I've been mystified by the apparently substantial nitrogen fixation that takes place in benthic systems, given the abundance of reduced nitrogen, during remineralization in sediments. Can you comment on why nitrogen fixation could be significant in a mud, for instance, an organic rich mud?
Yeah, this is a question that has eluded us for quite some time. Your question, I think, is like, if ammonium is an end product suppressor of nitrogen fixation, why are you getting such high rates in muds, right? Where you're getting,
Yes,
yeah.
that's
I don't think that question has been adequately addressed. I think one answer would be, well, it's patchy on even a micro scale, so that, end fixation is not suppressed on the micron level. There have been other hypotheses, some of which are interesting, but I don't think they've been proven. And that is that nitrogen fixation might actually be a way of getting rid of reducing power. You got nitrogen around, you got lots of nitrogen around. If you run out of everything else, okay, let's fix nitrogen. So it's not actually used as a nitrogen source, it's just used as an electron dump. And then there are folks around, I remember having this conversation with Ron Ormland, bless his heart, where Ron hypothesized that the enzyme for nitrogen fixation actually originated in many billions of years ago, and it was actually used to detoxify acetylene and convert it to ethylene. In other words, there was plenty of nitrogen around. This enzyme just evolved to get rid of a toxic triple bond compound in the sort of Precambrian environment. That's an interesting hypothesis, and someday some smart graduate student ought to jump on that one.
Yeah, that's
so it was like Ron, I remember, called it acetylene reductase.
That's great. I love that. Okay. Hey, thanks, Hans.
Okay, Malcolm.
Oh, hi. Thanks, Han, for a very interesting talk. I just have a very practical kind of question. In the next village to Stony Brook, there is a historic duck pond called the Setauket Duck Pond. And this was created, I don't know, maybe a long time ago by building a dam. And now it's a beautiful pond and it's surrounded by historic houses. And then as the community has developed, the watershed is surrounded by houses, all of which are on cesspools. And there is also in the watershed probably about a 12-acre protected agricultural area, which the local residents like to blame for all the eutrophication. But every summer now, and vice versa, but every summer now, this pond becomes highly vegetated. I don't know what all the constituent species are, but some are rooted plants. And you can almost walk on that water, you can guarantee that. The ironic thing is that our state legislator, who's chair of the State Assembly Environment Committee, lives right next to this pond. And so there's been a great debate about what to do about this. And there's been some expensive fundraising. Some say, well, we got to deepen the pond. And some say, well, that won't work. But I'm just wondering, as a practical thing, wouldn't one solution be just to harvest the biomass? I've seen these little skimmer boats in Florida lakes and ponds where you basically go around like a lawnmower and you skim off the biomass, take it out of there. It could be used for compost elsewhere, but I don't see as a practical matter how they're going to solve this problem, because it's never going to be sewered.
Well, if it ain't good, this sounds so much like the situation, Chris, that you invited me to participate in the Hamptons.
Very similar.
If they're not going to do anything about reducing nutrient inputs, I think it's pointless to do some of these other strategies. I think it's fine to remove biomass as long as you're also reducing the nutrient inputs. But one thing about removing macrophyte biomass is you might actually encourage the growth of planktonic blooms because the phytoplankton will become more competitive, particularly the cyanobacteria. So you need to be careful even about harvesting macrophytes in those kinds of situations. But the bottom line is there's no point going anywhere with this unless you can reduce the nutrient inputs at the same time.
Well, they talk about slowing it down by putting more advanced treatment, but that's not, I don't know if that'll work or not, but what do you think of the idea of doubling the depth?
Well,
basically emptying the lake and just taking out all the accumulated sediments.
well, yeah, well, I mean, dredging, they could dredge it, but if you dredge it, you got to take the dredge spoil and move it out of the watershed.
Yeah, that's for sure.
That's been proposed for Tahoe, and the Chinese actually went ahead and did that in some of the bays. And it actually, all it did was just put another loop in the nutrient cycle, because what they did was they put it up on the hillside and, they just put another loop in the cycle, basically. Nutrients going into the lake. So if you dredge, if you don't care about what's living on the bottom, and you dredge, move it out of the watershed, and then at the same time come up with a comprehensive nutrient input management strategy, that's probably your best, approach. But again, you need to be careful about removing the macrophytes and not inducing a planktonic bloom.
Yeah.
If you have enough nutrients still left in the system.
Well, lots of nutrients. Well, thank you.
Okay, I think, Jim, we're going to make you our last question, and then.
Well,
So
Jim's
go ahead,
probably
Jim.
gonna stump me over there, I think.
Yeah, I don't know about that. Great talk, Hans, as usual. People are getting more concerned about detecting microcystin in estuaries and coastal waters these days.
Yeah.
And my assumption had always been that it was coming downstream from tributaries, from lakes and stuff. But what you're suggesting is that maybe more of it's now, the source might be closer to the estuary than we thought before.
Well, especially if you're getting more extreme events that are leading to different hydrologic conditions, particularly freshening events in the upper estuarine areas. We're seeing that in North Carolina in several places, the Pamlico River, the Chowan, and Albemarle Sound. And in Florida, it's happening too. So I think both can occur, Jim. The toxicity due to lysis, and then
Right.
And then the toxicity due to in situ production.
I mean,
Another place is the San Francisco Bay Delta. That's another good example. Although there, the cyanobacteria are increasing in the lower delta because they have manipulated the water residence time so much and withdrawn so much water that they basically have turned these tracks, which are they're essentially diked off areas. They've turned them into cyanobacteria incubators. And then if you get a wet winter, which it looks like they might be getting in California, then all that stuff would be washed downstream.
yeah. Okay, thanks.
Okay. Well, that was great, Hans. So glad we could do that. And we're very grateful that you were able to join us. And again, I had sent you, you're getting lots of virtual applause there. And I sent you a link for a student meeting, and we can jump to that now. And everybody attending, I appreciate you joining in. And
Okay. Chris, did you send that to me by email? I can go back and check.
I did, yeah.
Okay.
It should be
All
in
right.
your inbox from this morning, I believe.
Okay. All right. Well, thanks, everybody. It's too bad I can't, party with you in person. I'll welcome you all to my porch over here in Beaufort at some point, and we'll get together and party.
Very good. Thanks so much, Hans, and have a great weekend, SoMAS.
Okay.
Best wishes.